Hi everyone, today let us see about types of cell cultures. That is, in the case of animal cell line, we can see the types of cell cultures. Okay, this animal tissue culture can be divided into two categories. That is, the cultures that allow cell-to-cell interactions and encourage communication or signaling between the cells. That is, the cells which allow interactions or communication, the cells in which communications happen between the cell to cell, that is one type. And where the interactions or the signaling is not allowed or missed, that is another type. So cultures that allow cell-to-cell interaction are type, and cultures in which cell-to-cell communication or interactions are lost is one type. So the first category can be classified into three types, that is organ culture, histotypic cultures and organotypic cultures. The second category includes monolayer cultures and suspension culture. So the culture in which communication is allowed can be divided into three types, that is organ culture, histotypic culture and organotypic culture. In which communication is not allowed can be categorized into monolayer culture and suspension culture. So first let us see about the first category, that is organ culture, histotypic culture and organotypic cultures. Organ culture, as you know, culturing the organs, whole organ or the tissues or cells taken from the particular organ can be culture to form an organ. So organ culture is a culture of native tissues that retains most of the in vivo histological characteristics, whereas that is most of the histological characteristics means all the characters of the animal particular organ, that is particular organ of the particular animal should be symbolized or established. That is organ culture. But when culturing the cells for reaggregation, that is to reaggregate the cells, if you are going to culture, that is called as histotypic culture. So organ culture is a culture of native tissues that retains most of the in vivo histological characteristics, whereas culturing the cells for the reaggregation, that is to yield tissue-like structure, is called as histotypic culture. Also in histotypic culture, you can say individual cell lineages are initially derived from an organ and then cultured separately to high density in a 3D matrix. That is to study interactions, signaling between the homologous, that is interactions, signaling systems can be studied with the help of this histotypic culture. Also, while doing organ cultures, you can use whole embryonic organs or small tissue fragment in vitro, can be cultured in vitro, so that they can retain their tissue architecture, that is the characteristics distribution of various cell types in the given organ. Then the last one, organotypic culture. Organotypic culture, you can mix different forms of cells. So different origins of cells can be mixed together in specific proportions and in spatial relationship, so that you can reform a component of one particular organ. So you are going to introduce something, invent something new, that is you can mix different types of cells and you can come with the final product. So the recombination of different cell types to yield a more defined tissue or organ is called as organotypic culture. And cell culture, as you know, it's a process of removing cells from an animal or plant and their subsequent growth in an artificially controlled environment. So commonly cell culture, we will be using the term. Most of the people don't know what do you mean by the cell culture. Cell culture is nothing but the process of removing the cells, maybe from an animal or from a plant, and then culturing the same cells in an artificially controlled environment, that is called as cell culture. This cell culture is broadly classified into two types, that is primary cell culture and secondary cell culture. We can discuss about this primary cell culture and secondary cell culture now. So the primary cell culture, this is the first culture, that is a freshly isolated cell culture, or the culture which is directly obtained from animal or human tissue, or maybe plant tissue also. So the tissues that can be obtained from animal or a human tissue by enzymatic degradation method or mechanical methods. Enzymatic methods is more commonly used. Mechanical methods you want to crush, some of the mechanical methods you have to use. But in enzymatic, just by adding trypsin for some of the enzymes can be added, and you can separate the tissues, a particular tissue of interest, and you can go for the primary cell culture. So freshly isolated cell culture that being cultured is called as primary culture, cell culture. So these cells are typically slow growing. These primary cell culture cells are typically slow growing one. It is heterogeneous and carry all the features of the tissues of the origin. The primary objective of this culture is to maintain the growth of cells on an appropriate substrate. That is the main aim of the primary culture, is to maintain the growth of cells on an appropriate substrate available in the form of glass or plastic containers or etc. Under environmental, that is controlled environmental conditions, you can store or preserve the particular cell types. Since they are directly obtained from original tissue, they have the same karyotypic characters. That is number and appearance of chromosomes in the nucleus of eukaryotic cells will be the same as the original tissue. Okay, once subcultured, primary subcultures can give rise to cell lines, which may either die after several subcultures. That is called as finite cell lines. So what do you mean by finite cell lines? On subculturing, the primary cell cultures can give rise to cell lines, but it can die after several subcultures. That is called as finite. If it didn't die, but it continues to grow indefinitely, that is called as continuous cell lines. Usually happens in the cancerous cells. So usually normal tissues give rise to only finite cell lines, but in the case of cancerous cells or tissues, you can see continuous cell lines. That is called as aneuploid condition. Okay. So if you are going to use a normal tissue or normal organ, from that you are getting some cells and you are going for culturing, cell culturing. After several subcultures, you can see the growth may stop. The cell lines will stop further dividing. So that is called as finite cell lines. But if it continues to grow indefinitely, that is cancerous cells. But some exceptional cases are there. Like MDCK from the tissues from MDCK, dog kidney, fibroblast, T3, etc. So these are some of the examples here. And now with the help of new technologies, due to the emergence of newer technologies, that is 3D culture, the use of primary cells is being increasingly prevalent and achieving improved results. That is more wonderful results are being obtained using this 3D culture. And also these primary cells, which are directly obtained from human or animal tissue using enzymatic and this or mechanical procedures, can also be classified into two types, that is anchorage dependent and anchorage non-dependent. This anchorage dependent can also be called as adherent cells. Adherent cells are nothing but the cells which require attachment for growth. That is anchorage dependent or adherent cells. These cells are capable of attaching to the surface of culture vessel and then it can be cultured. These types of cells are often derived from tissues or organs, that is from kidney, where the cells are immobile and embedded in connective tissue. So these things are all come under anchorage dependent or adherent cells. The next one is anchorage independent. As a name suggests, there is no attachment is needed. This can also be called as suspension cells. Suspension cells do not require attachment or any support for their growth, and also called as anchorage independent cells. Or suspension cells are isolated from the blood system. For example, white blood cells, lymphocytes, plasma. These are some of the examples of this anchorage independent cells. So this can be cultivated using suspension culture itself. There is no anchorage dependency, so no need to use any basis with having attachments for the growth of animal cells. And also, for several reasons, the primary cultures have a limited lifespan. That is, the cells cannot be maintained indefinitely. Also an increase in cell numbers in primary culture can result in exhaustion of the substrate and nutrients. So it should be in the controlled form only. If it leads to inhibition of cell growth, means that it will be called as confluence stage. So due to when you are culturing primary cell lines, due to some an increase in cell culture and also exhaustion of substrate and nutrients, this may lead to death of cells, and this stage is called as confluence stage. So confluence stage, here the secondary culture or subculture needs to be established to ensure continuous cell growth. That is, again you have to add the nutrients for the growth of animal cells, or you have to carry out some other process. Now let us see about secondary cell culture. This secondary cell culture simply refers to the first passaging of cells, and which is the switch to a different kind of culture system. That is, first you have to obtain the culture from the primary culture, and then you have to carry out some this is usually carried out using adherent cell cultures that occupy all the available substrate, or when cells in suspension culture surpass the capacity of medium to support further growth and cell proliferation begins to decrease or ceases completely. So at this stage, secondary cell culture is done. So we have to maintain the primary cell culture. That is, primary cell culture, as I have already mentioned, it has a limited lifespan only. But you have to maintain the cell culture for the longer part. So you are going for the secondary cell culture now. So as to maintain optimal cell density. Clearly note the point. So as to maintain optimal cell density for continued growth and to encourage further proliferation, the primary culture has to be subcultured. So I think you understand. So as to maintain the optimal cell density for continued growth and to encourage further proliferation, the primary culture has to be subcultured. So this process is called a secondary cell culture. You may have the doubt, why I have to switch over to secondary cell culture while I am having my primary cell culture. Primary cell culture can stop within a stage. So it should not be stopped. You have to maintain the optimal cell density. So for that purpose, you are going for the secondary cell culture. You can see the difference between primary cell culture and also the secondary cell culture in the following table. So primary cell culture, as you know, it's directly obtained from animal or plant tissue, but secondary cell culture, it originates from a primary cell culture. And primary cell culture, you can see closely resembles the parental tissue, but the secondary cell culture, some deviations may occur because we are maintaining environmental conditions, but also there may be some changes. So that's not closely resemble the parental tissue. Also, the biological response of the cell may be closer to that in vivo environment, but the biological response of the cell differs from that in vivo environment. And the first culture is derived from original cells or tissues, but here from an existing culture. In the secondary cell culture, you have to derive from an existing culture, that is from the primary culture. And also primary culture cannot be transformed, but secondary culture can be transformed. And in primary, you can see only less chance of mutation, but an increased chance of mutation or genetic alteration of primary cells is seen in secondary cells, acquired through steps of rinsing, dissection, and mechanical or enzymatic disaggregation. That is, from the original organ or tissues, you have to get the cells, means you have to go through these steps: the rinsing, dissection, mechanical or enzymatic disaggregation methodologies. But in the case of secondary cell culture, you are getting the primary culture you are getting the cells from primary culture. So if the primary culture is an adherent culture, the first step is to detach the cells from the attachment. From the attachment, you have to detach the cells, and you have to use for the further experiments. This can be done by mechanical or enzymatic means. Then the cells have to be detached from each other to form a single cell suspension. And also here in a primary cell culture, you can find a finite lifespan, but prolonged lifespan of cells can be seen in secondary culture, periodic subculture. This is maintained by periodic subculturing. Also, this periodic subculturing can produce immortal cells through transformation or genetic alteration of primary cells. And in primary culture, the risk of contamination is high, more difficult to maintain this primary culture because it is directly taken from the human or animal tissues. So it's very difficult to maintain. But due to some of the conditions or stress the cells are undergoing, so it maintains to in better condition, so it can be easy to maintain the secondary stage. The risk of contamination is lower and also comparatively easy to maintain when you compare to primary culture. So these are all the all about the types of cell culture. I hope you all enjoy the video and also you understood what are the types of cell culture. If you like the video, kindly like, comment, share and subscribe the video. Thank you.